So my topic tonight is what's called epigenetics. And as you may know, epi is up on, right, or above. And obviously you know genetics. This is actually an amazing discovery in the last decade or so. And it's truly revolutionizing almost every field. Certainly it's revolutionizing medicine. And my subtitle here is How Lifestyle Controls Gene Expression. And when you think about it, ever since Mendel discovered genes, inheritance in genes, we've wanted to control it. Right? I mean, if you think about it, you too, you maybe never verbalized it that way, but we all want to be able to control the expression of our genes, because unfortunately, or fortunately, we can't choose our parents. We can't choose our genes. But it's, as you're going to find out, that's a very minor obstacle. It's a minor obstacle. Yeah, for those who are interested, there's some of my pedigree there. Okay, so I give this talk to lots of different audiences, and don't be intimidated, but I'm going to go ahead and do it the formal way, where there's some actual learning objectives and so forth, okay? So what you should be able to do to finish this presentation is be able to define genetics and epigenetics, and in particular understand the difference between them. You should be able to recall 3 milestones in genomics research, okay? Believe me, these are really easy. Don't get scared. And this is probably the most important. You should be able to explain your genes are not your destiny. This is a common, all too common misconception that genes are destiny. But of course, as a physician, you would expect me to first have a couple of case histories, right? So we'll talk about a couple of cases, if you will, first. And the first one is thanks to you. I had the privilege of helping spend $2 million in the Marshall Islands. So, in fact, they call the Marshall Islands the Pearl of the Pacific. And so it's quite pretty over there, it's true. And what we were doing, though, is there's problems in paradise. There were 3 diabetics in all of the Marshall Islands right after World War II, when the survey was done of the Pacific after the war ended. 3 diabetics. Now, over 35% of the population has got diabetes. The average blood sugar in a survey done in the capital city was pre-diabetic. The average person was pre-diabetic in the Marshall Islands. So, and ever since the war, the U.S. government has what's the word I want to use? Anyway, they were a protectorate for a while, and so on. So we have a vested interest in their health. So we spent a couple $1 million doing a study to see how lifestyle change could change this epidemic of diabetes. And so here's some of the pictures. Again, we both apologize that the lighting, if we turned out all the lights, the camera wouldn't work well, and probably some folk who needed more sleep last night wouldn't get much out of the talk. But hopefully you can see enough to get it. But this was our medicine. Have you ever heard the expression, let your food be your medicine? Well, in this research, that was the truth. That was our medicine. Well, I call this our compounding pharmacy. Anyway, so the kitchen is our compounding pharmacy. And we were teaching the people how to eat differently and in ways that they like. The other thing you see here is a little exercise room. We did have a little exercise room, but the best exercise is not the kind you do at the gym. The best exercise is the kind you can do any time of the day that you want to. So generally it's walking. Of course, if you live in the Marshall Islands, swimming is almost as available as walking there. And here was the result that we published paper of our results. This was a 24-week intervention. So it's about 6 months. And this was the treatments compared to control. So this is really the average was 10.2 A1C. If you're not diabetic, you wouldn't know what this means. But the normal range should be under 7, and we're not even showing 7. Okay, these people they're so diabetic that nobody has a normal number. Anyway, so this is way off the scale, so to speak. And in fact, the test for the test for A1C only goes to 14. So, we had people that we didn't even have a readable number. They were just off the scale. But with 7 weeks, was the average time of the second test here, and they had dropped remarkably. And then over the next follow-up period, there was some bouncing back a bit. But still, the end result was over 1 point difference, and that, to put it in perspective, using medication, you often have to use 2 medications to get that much of a long-term change. Anyway, so this was quite remarkable. In fact, when I've shown this to some medical audiences, and I told them really what we were doing, the question is asked, well, wow, that's great. What new treatment are you using? You use an injection pump. What did you do? And because we all in medicine think of medicine. But in this case, we're using lifestyle. Here's a beautiful illustration I like. This is the first group of patients we had the first 10 days. And this is sort of a regression line here showing that their average finger sticks. If you know anything about regression, they lost 74. They dropped 74 points in their blood sugars, morning blood sugars in 2 weeks. And this is absolutely remarkable that you can have that kind of change. And so what I'm hoping I'm doing with these case histories is I hope that I'm creating in your mind a question, well, how could that be? How can this happen? And so my second case study is taken from my lifestyle center. I used to live in California a few times. I've lived here 2 or 3 times, but I'm actually from Virginia, and I can't stay away from Virginia for too long. So I actually have moved back, and this is our lifestyle center, my practice, if you will. I'm a preventive medicine and lifestyle medicine doctor. Matt didn't have a chance to tell you anything much about that, but that's okay. And we have we use something called the Complete Health Improvement Program as the curriculum for a lifestyle intervention program that we have there. In fact, my wife couldn't be with me on this trip because she's there making sure that things keep running while I'm away for 3 weeks helping out here. Now, some of you have seen this kind of a study before, an echocardiogram. This is where we use ultrasound to visualize, the heart, and it is a living. You can do it in full motion. So this is a typical echocardiogram. You get this 3D picture, and then you get some of these cross sections. And what we use it for, as you may know if you've ever had one, we use it to look at the functioning of the heart, and in particular to see if there's a leak in the valves, or is the heart working properly. So I just use that as an illustration to tell you that's the kind of study that was done for this particular patient. Mr. Kelly had a study done in 2006. And here's what they did after taking this. It says, after reviewing everything we recommended, he undergo coronary angiography with intervention if indicated. And do you know what that means? That means basically the doctor was saying you need to have an angioplasty and a stent because you've got some kind of poor wall motion. Your heart's not working as it should. Then 2 years later, 2008, he had another study done. And what had happened in between was he had made some lifestyle changes of his own, and then he wasn't getting enough improvement, so he came to our lifestyle center for an 8-week program. And here's what the results showed. The echo portion of the test was considered negative for ischemia. Patient has done well with lifestyle changes. Planned he will continue with diet and exercise and aspirin. Now, one of the things that was interesting about this was that the cardiologist who did this, Dr. Austin, who did it there in Virginia, the first study was actually done in, I think, Missouri. But the one in here, Dr. Austin said, maybe you didn't actually have heart disease. He had never seen a 60-some-year-old male have improvement in ischemic heart disease. It doesn't happen. So it does happen, but to him, he'd never seen it. And so he actually said in his formal report there was a possibility this was artifactual, meaning it wasn't really there, it was just an aberration on the study. Of course, the first doctor, Dr. Spady, would have been a little upset with that idea that he had misdiagnosed it. Now, I want you to contrast that with this. Here's a study that was published in the New England Journal of Medicine in 2009. And this was a study that was done by and published for surgeons. Okay? And what they were looking at was, I don't know if you realize it, but when they do a bypass surgery, they take a vein out of your leg and they stitch it into the circulation in your heart to bypass clogged arteries. And, you know, everybody's trying to reduce the cost of medical care because it's just skyrocketing and it's breaking the country. And so what they're doing is increasingly the insurers are putting pressure on surgeons for their surgeries to last longer. And so the surgeons are trying to study and figure out, okay, what can we do to make this operation so it lasts longer? I think that's all good. Everything so far, all that's really good, right? But, well, I mean, you don't want to have these things every week. So anyway, so they did a study, and what they had found was that there's two ways of harvesting the vein out of your leg, and they wanted to compare and see which way they harvest that vein. Does it make any difference in how long the vein will last? That makes good sense. So they did a study, and there's two ways of getting the vein. One is called open excision, and the other is laparoscopic. And you know, laparoscopic is where you make a few little holes and you stick the thing in there. And so if they can pull your gallbladder out of your abdomen with a little hole, they can pull that vein out of your leg. But you might understand it's a little hard on the vein. So, and you're not throwing it away. You're going to still sew it in there. So what they did was they looked at this, and they were looking at how many of the surgeries failed within how long? Yeah, 12. So they were looking at how many failed in 12 to 18 months. Yeah, I know some of you are like me, are already, can't believe it. I would have thought they were looking at 6 to 8 years. Anyway, but if they took it open, cut the leg open, it was only 38% that failed in 12 to 18 months. And they were pretty happy about that, because if you took it with the laparoscopic, it was 47% failed in 12 to 18 months. And so the average overall was 43%. Okay, so the surgeons were quite impressed. They looked at this and said, we need to use open excision to get those veins. Now, I'm a lifestyle medicine doctor, and I looked at this and I said, we should not do these things. They last only 12 to 18 months. And so, and here is a contrast, is a case using lifestyle to treat the exact same disease. Okay? I'm going to show you a bigger picture. Some of you who are familiar with Dr. Esselstyn's book on how to prevent reverse heart disease, this is the famous picture of Dr. Crow, who was the surgeon at Cleveland Clinic. He's still actually working at Cleveland Clinic. He had a study that showed this lumpy bump. You can hardly see the boundaries. I'm sorry for the light. But over here, hopefully you can see it better. It's nice after 32 months. And what was his treatment? He had compounding pharmacy, plant-based diet. So after 32 months on plant-based diet, and he could not take medication. He was allergic to the medication, couldn't take it. This is what he had. He's excited. So, to me, that's quite a contrast. Get that vein out of your leg, 43% fail in 12 to 18 months. But if you make a lifestyle change, you can have a completely open, clear artery. Quite amazing. Okay, so that's my cases. Now let's get to the real talk. Every time I share this kind of thing, I always have at least someone, and you don't hold up your hand, but at least someone in the audience that says, you don't understand, Dr. Kelly, I've just got bad genes. I just got bad genes. That's my problem. And you know, it's true, we do inherit our genes, and genes do have an impact on health. No doubt about that. But before I go in, let me just tell you something while your mind is still fresh, not filled with all these details in a minute I'm going to share, but what else do we inherit besides our genes, our DNA, from our parents? We inherit taste. I tell you, of course, I'm an old guy, and this may have changed a bit in the last 40, 50, 100 years, but when I was young, I can still remember not a few guys who would say to their wife, "Hon, could you just learn to make that cornbread the way Mom used to?" So some of you ladies will laugh at that, but because you that's right. We grow up liking something a certain way, and a lot of things we grew up liking a certain way. So yeah, we inherited a lifestyle. In fact, in many cases, it was somewhat forcibly pushed upon us. I can remember a few things I was forced to eat that I really didn't care for. So that was a sort of, right? That was changing my life. Well, in getting old now, what was that? January 2010, I think. Time Magazine had a whole issue on why your DNA isn't your destiny, and it was talking about the new science of epigenetics reveals how the choices we make, that's our lifestyle, can change our genes. Change our genes, and those of our kids too. Now, when some people first saw that, they said, change our genes. Wait a minute, I think you can't change your genes. Yes, you can. You cannot change your DNA sequence, but you can change your genes, and I'm going to show you that. This is what's so amazing. And how about that? Those of our kids. Now, of course, if you're not having any more kids, you won't be able to change the ones you've already got. But you can actually change the genes of your kids. I'll show you. This is amazing. Okay, a little history. This is an actual picture of Watson and Crick receiving the Nobel Prize for their wonderful discovery of deoxyribonucleic acid, DNA, as the molecule of inheritance. And this is actually a little pictogram. It's not a picture, but it illustrates that, you know, this is a double-stranded molecule. You've heard that, sort of a helix. Did you know that the two, if you think of this as a ladder, the two sides of the ladder don't touch each other? Did you know these two molecules, strands, do not actually physically touch? There's a little gap. Do you see that little gap here? There's a gap. And they are just held together by hydrophobic forces. It's if you're into chemistry, like some of us are, it's really elegant. It's beautiful. But anyway, so this silent embrace. Now, the Human Genome Project, which finished about a decade, 2010 or so, Dr. Francis Collins, now head of the NIH, was the head of that particular project. And what they found when they sequenced the human genome is 3 billion of these base pairs. So looking at this there's 3 billion of these rungs in the ladder. 3 billion crosses between these two in every human cell. Not in a human being. 3 billion in each cell. Did you know there's enough DNA if you took it out of your body and put it end to end, could reach to the sun and back? Each human. Over 25,000 individual genes. Now, to us, that you think, wow, that's a bunch. Actually, science was amazed that it's so few. Some other animals have more genes than humans. That boggles the mind a bit, because they thought that, well, the more complicated and more higher, I guess, a higher life form should have more genes. Millions of variations, okay? And some of them cause a disease. Some genetic variations cause a disease, most don't. Now, the way that most of us are familiar with this DNA variation is that's what a DNA test is all about. That's how, when you get a DNA test and you can tell who done it, it's based on these millions of variations that occur. There are more variations than in the fingerprint, so it's a more accurate way to tell people apart than even the fingerprints are. Now, I mentioned that some of them cause disease. One illustration is something called sickle cell disease. Sickle cell disease, here's a sickled cell. Here's the way a normal red blood cell should look. And as you might imagine, when the cell gets in this kind of a shape, it doesn't circulate well, it doesn't function well, and it causes a lack of oxygen to get to the tissue. So if you're not familiar with this disease, anyone who is can tell you right away the main symptom of this is terrible pain, excruciating pain when the circulation doesn't work like it should. Well, that's all because of a single mistake or a single difference in the DNA that causes this condition. One difference, base pair. There's a T or an A. So the sequence is GTG when it ought to be GAG. That's for glutamine, thymine, glutamine instead of glutamine. Oh, guanine. I'm sorry, guanine, not glutamine. I'm getting mixed up with proteins. Anyway, so this one thing causes sickle cell, but the vast majority don't cause it. Okay, most of those variations don't cause it. All right, next point. Anybody below the age of 15 can tell me what these are? Any of the younger ones in the audience? How many we got? What are they? Very good. Exactly. These are hedgehogs. And there's 3 of them. The mama hen triplets. And I want you to think for a minute about your basic biology. You probably learned this in high school. Where did each one of these little creatures come from? Well, an egg from mom met up with a sperm from dad, created, and they joined together, called fertilization, became a single cell, right? And this was a complete cell because the egg only had half of the chromosomes, right, to make this little creature. The sperm only had the other half. But when they got together, now there was a full set. Okay, same place, by the way, where we came from. And then that one cell began to grow and reproduce. So one cell became 2 cells. Each one of those became 2, so they became 4, so on and so on and so on, right? Well, in the case of a human being, 9 months later and trillions of cells later, you were born. Okay, so every cell in each one of these, every cell has a copy of the first DNA, right? That first set of chromosomes. They were just copied each time. So every cell in your body has the same genes, the same DNA. Okay. Now here's my question for you. First question: if every cell has a copy of the same DNA, then how does it produce such different tissue? How does it? Some of it make an ear, some of it a toe, some makes a brain. It's a real, obvious question, and for the longest time we really never thought about the implications. What does that tell us? It tells us that the majority, well, first off, that all those genes have to have switches. And the second thing it tells us is that in every cell in your body, most of the genes are turned off. See, I've got all the genes in my ear for a liver. I'm really glad they're turned off. And in fact, there's all the genes in my right ear for a left ear. I'm also glad those are turned off. So when you think about it, most of the 25,000 genes in each cell are selectively turned off. And what's exciting, and I'm not going to go into this right today's talk, but there's a whole growing field of how we're learning to turn the gene switches on or off in living cells. We'll talk about that another time. Because they're actually turning skin cells into cardiac cells. We're learning how to turn a living cell from your skin into a living cardiac cell that will beat and act like a heart cell. Yeah, it's amazing. But it's all about the switch settings. See, like we have a room here. The lights are on. If we had the right kind of switches instead of, unfortunately, only one switch, but if we had enough switches, do you realize we could turn the lights on in different patterns and make the room look quite different, right? We could darken the front, or we could darken one corner. Well, in your cell, you have all these switches. You can turn the cell by how you set the switches from either a skin cell or a cardiac cell. Now, this is not so amazing when you think about it, because what Dr. Whitelaw here, an expert on this, says is, we don't really inherit DNA. What we inherit from our parents are chromosomes, and chromosomes are only 50% DNA. The other 50% is made up of protein molecules, and these proteins carry the epigenetic marks, or what I'm calling switches. Okay? And I'm going to illustrate this right here. You know what monozygotic twins, commonly called identical twins, they come from, this is when that first cell that was created from the egg and the sperm, if that one cell, instead of just producing one organism, it can actually have a variety of things, can cause it to split into 2 or 3 pieces, and each piece will grow into a full human being or a full animal. So that's what a monozygotic or an identical twin is, a case where the developing blastula or morula, what they call this thing during embryosis, if it splits into two parts, each one will grow into a complete human being. Okay, but what do you now know about those two human beings? They are clones. They both have the exact same DNA. They both have the same genes. They are truly clones. I mean, we've gotten to where we don't like that idea. We're like cloning human beings. Well, actually, we've been cloning human beings in the womb ever since, Adam and Eve. But we didn't realize it, perhaps. So what I want to show you is what the nice thing about monozygotic twins is you can do remarkable studies on people to look at the way non-genetic differences affect you, right? Because these two people both have the same genes. And of course, because of the way things happen in our world, not always so nice, sometimes one twin will have never known the other twin. That happens. Now that's, from a family standpoint, I think that's bad. But from a science standpoint, it makes a remarkable time to do some studies on how environment affects genes. So here's what they did. They looked at, this was published in 2005, Proceedings of the National Academy of Science, and they were looking at the differences in epigenetics of 3-year-old versus 50-year-old. And what this is twin A and twin B, and you can see that there's not much difference in these bands, they call them, of the DNA, but by the time you get over here to a 50-year-old, you can see definite differences. You see how much more prominent the differences are here, the color of these bands versus over here. And so what they found was there was thousands of differences more by the time identical twins were 50 years old than there were when they were 3-year-olds. Now what does that tell us? It's telling us that during their lifetime, from age 3 to age 50, there's tremendous changes in the gene switches between these two twins. Well, that's for the ones that like chemistry. So let's go here and look at this. This was the Journal of the American Medical Association 2008 by Dr. Feinberg from Johns Hopkins, one of the real experts on this. And what he's showing here, let me just break this little diagram down. This is a chromosome. You kind of recognize that, right? That you remember they're kind of like X-shaped. And then that is made up of something called chromatin, which is a sort of a coiled DNA. And then on the DNA, you can have these methyl groups. A methyl is a carbon with some hydrogens. And if that gets attached to a particular part of the DNA, we have learned that constitutes, you can change the switch setting just by putting a methyl group on the certain places on the DNA. And then there's another thing. This DNA is wrapped around something called histones. These are proteins, and it looks like a pill. It looks like a pill that you could cut into 4 parts, but it's a 4-part protein, and it's what it's supposed to be. And can you imagine if you're trying to transcribe this part of the DNA, when it's coiled up like that, you can't get to it. Well, if a epigenetic factor gets attached to this little tail on that protein, it actually causes the thing to unravel enough that this section of DNA is now available. So in essence, over here is turned off, over here is turned on. And all it takes is this epigenetic factor to be attached to the histone protein. So these are just 2 of the ways the switches work. Okay, there's lots of other switches we won't have time to go into. So it turns out that the DNA and the chaperone proteins together determine which genes are active and which ones are not, which ones are turned on, which ones are turned off. And what we've discovered in looking at how to turn these on and off is that there's an amazing 4-letter word that describes the most powerful effect on these switches. And it's diet, or food. I like to think of it. I like food more than diet. Anyway, it turns out that food exerts the strongest effect on these switches of anything that has so far been discovered. I'm looking at the clock and I'm thinking, I'm going to skip this one. Some of you have heard about this, so I won't go into great detail, but there's studies that have looked at the fact that the availability of food during adolescent years has an impact on the susceptibility of the grandchildren to heart disease and diabetes. I'm not going to go into it. I'm just telling you the summary, the substance of it. This was published in 2007 and a few other articles. But basically, they had a 10-year difference. They had a 10-year difference in life expectancy in grandchildren that could be traced back to the abundance of food during the adolescent, pubescent years in the grandparents. This was some records they had in Sweden. Yes.
If I understand correctly, the scary part about that is that's how far they tested. They don't know if they
Yeah, that's a great point. That is, I don't know if that's scary or exciting, but I could argue that. But anyway, yes, it's very, very
depends on whether your great grandparents had a good diet or not.
You know, the way I'm quite pragmatic. It's like I can't really change what my grandparents or great grandparents' diet was or available. But I appreciate your point because actually that is you see, she's right. We're looking back at 2 generations. I'm pretty impressed that they could even do that. You realize in America we can never do a study like that. We don't have records adequate to track that kind of thing down. But in I believe it was in Finland or Sweden they did that. Finland, I guess. All right. Here's and here's what they found. This is the bottom line conclusion: that the transgenerational responses, transgeneration meaning from one generation to the other, that nutrition was the main influence on longevity. Okay, that was the conclusion here in the European Journal of Human Genetics, 2007. And the nutrition, as I said, switches genes on and off, and diet exerts the strongest effect. Now, let me ask you something. If I were to talk slow enough or stop talking so you could think a minute, what would be one of the most significant points about that? If your diet and your ancestors' diet is one of the strongest influences on the gene switch settings, well, you know what I think is, hey, I changed my diet. I can change my diet. I have control over my diet. Now, it's true that there are times that certain foods seem to have control over me, but I gained the victory eventually. You know, I used to like to eat a lot of things that I don't choose to eat now. So what I'm getting at is this is empowering. You can change. Diet is under your control. Okay. All right, we talked about, there's a whole field called nutrigenomics, by the way, building around this idea of trying to develop and design nutrients to turn on and off certain switches. It's going pretty slowly, to be honest with you. It's a whole lot more complicated than they thought it would be. And when we start life, we're going to turn a corner here a little bit on another point now. When we start life at birth, or we might argue differently, but anyway, whenever it starts, we start with the gene switches are set by the environmental influences upon our ancestors. Okay? So at birth or conception or whenever life starts, it turns out that we now realize that I not only inherit the DNA from my parents, okay, but that DNA has switches, and the switches are either on or off. The switches don't have a neutral setting. So that, whether they were on or off when you were born, was largely determined, as I'm going to show you, by things that happened before you had any control over it. Okay? So there's a little study I'm going to show you. You've probably heard of this. If you've seen any of this, this is one of the most famous ones, the agouti mouse. And it turns out that this mouse over here is worth thousands of dollars, hundreds of thousands of dollars. Because this mouse, and this is just a regular old mouse you might catch in a mouse trap and throw away. But this one, don't throw away. We want to resuscitate it if it gets caught in a trap. Because we have engineered it to have genes to cause it to get the diseases that are killing humans. 3 out of 4 human beings in America are being killed by lifestyle diseases. The agouti mouse has been engineered to get those diseases so that we can study it. Okay? And they're studying agouti, all kinds of things in agouti mice. But at Duke University, about 13 years ago, there was a kind of a wild idea that they had in the research there, and that was they wanted to use the agouti mouse to see if they could use nutrition to turn off those genes that had been put in there to cause disease. And of course, a lot of the scientists say this is craziness. Can't do that. But the researchers did it, and when they published their results, here's what they found. This is the mother, and this is her baby. And of course, you can tell that doesn't look like, remember I showed you the 2 regular mice. This mouse has none of the propensity to overweight, doesn't have the color of fur that goes along with those genes. These genes that are in this mother were silenced in this baby. And the baby was born with the genes turned off. And how did they turn them off? They fed the mother during pregnancy a special diet that is known to turn off the genes. It's called a methylating diet. You can read this article. It was actually published a long time ago, 1998. But this mouse is still making waves in science because it's so remarkable. What they proved there was that by changing the diet of the mother during pregnancy, you can change the genetic phenotype of the baby. In other words, if we were doing an examination, we were bringing mouse in and we were going to put the ones that had those genes on over here and the ones that had them off over here, and those two mouse came in the door, we would say they had different genes, right? They got different genes. Look at them. You can just tell it by looking at them. What's the matter with you? But they don't. DNA tests show they have the same genes. And here's what the author said. The effects of the special methylating diet were evident for 3 to 4 generations later. Yeah, now anybody here who reads the Bible at all recognizes that. So that's remarkable. That's from Exodus 20. But anyway, but for 3 to 4 generations, they could see the impact still evident of the diet that the mother had been fed. Yeah, so think of the implications. Anyway, this is exciting. To me, this is good news. Now, it's kind of bad news because of the way things are going in our country, but the potential, it could be great news. I got another study that'll really shake you up. Yeah, right here it is. This one is pretty, I didn't know your name, but I agree with you. This is pretty scary. This was published in the Journal of Neuroscience in 2008. Now, first thing I got to say to you, this is the Journal of Neuroscience, and what are we talking about? We're talking about diet. You know, you think, what are they studying diet for in neuroscience? Well, diet may have more impact on your brain and your ability of your neurons than you ever imagined. So here's a study. And this time what they're doing is they're taking a pregnant rat, not a mouse. And this is a regular old white rat, nothing special, no genetic modification. You know, if she gets caught in the trap, no problem. Don't have to resuscitate this one. And they're taking, and while she's pregnant, they're feeding her a high-fat diet, not a methylating diet. This is a high-fat diet. Yes?
What kind of fat?
This was a typical...
Was it hydrogenated fat like the problems
this was that's a great question, and I don't have the answer to that.
Because that would make the study be completely different, I would think.
Well, what I do know is, I have the article on my computer, and we can answer that question. We can look it right up here. I don't know the answer off the top of my head. I believe that it was your typical a combination of hydrogenated and non-hydrogenated and animal and plant fat. I don't really know. But I have looked up one thing that someone asked me one time, and I wondered about it myself, and that is how much fat. And I might as well, by the time I get to the end, I know you'll want to know that, so let me go ahead and tell you. It was around 55 to 60% of the calories from fat. Now, you may know that the recommendation is that we not have more than 30% of our calories from fat, and then the American average is 38 to 40%. But I happen to have looked at an interesting study I saw published from the Appalachian Mountains in a study they did in grade school children. And what they did was they were wondering what kind of a diet grade school children in Appalachia was eating. And so they did something called a subtraction analysis. They took the food that was served in the cafe line or the line, and they analyzed the fat content. And then they went after the meal and got the food out of the trash can, and they looked at how much fat was in what was thrown away and did a subtraction. It turns out that what our children are eating in grade school is around 55% of calories from fat. Think about that. So what they fed her a high-fat diet, and what they were able to do was trace the impact on the expression of a gene on chromosome 3 in this developing fetus, not the mother, the fetus. And this particular gene was a gene that caused the formation of brain cells, neurogenesis. It created more neurons. So the diet fed the mother. The first little interesting thing is it changed the anatomy of the brain of the baby. We can stop right there. That's pretty earth-shaking. So the mother's diet was changing the number of brain cells of a certain type that was produced in the developing baby. Now, this particular case, and the reason it's in neuroscience, is that those particular cells took up residence at the base of the brain where they are hormone-producing cells that happen to affect your appetite and your eating behavior. So it's hormonally active cells. The result was that in the offspring, offspring to weaning, weaning to adolescence, had higher blood lipids. That would be high cholesterol and triglycerides. Higher food intake. Supersize me. This baby really preferred supersize. Fat preference. This baby, compared to babies that the same mother had fed a normal diet, this one had a higher preference for fat, earlier onset of puberty, started reproducing at a younger age, higher body weight. And what was really kind of earth-shaking about when this was published in November 2008 was that almost every pediatrician in the audience was like, their little lights were blinking. This is exactly what we're seeing in pediatric practices today. And so in fact, I remember a comment that was published on this, I think in the Wall Street Journal, when the acting head of the American Heart Association commented on this study that was released. She said, there may be more to America's obesity epidemic than we have ever imagined, because the fast food places are feeding high-fat food. Our kids are eating it up. Well, some of us are too. And the result is we're seeing this kind of a profile. So it's very disturbing. But the good news is that very same mother fed a healthy diet had these changes were different. Okay, this didn't happen. Okay, I got to close. I'm running out of time here. This was published by Dean Ornish. Changes in prostate gene expression in men undergoing an intensive nutrition lifestyle intervention, proceeding in the National Academy of Science 2008. This was a study in 30 men who had prostate cancer. Now, prostate cancer is the leading cancer in males, and certainly the first world, increasingly around the globe. And it was a single-arm study, 30 men. They didn't have surgery or radiation treatment. They received a lifestyle intervention program. And the reason probably this was allowed to be done by the ethics study was the ethics committee was these men had indolent, so-called low-risk prostate cancer. So they weren't expected to be killed by this cancer. But it's nonetheless serious. I certainly wouldn't want to have it. And so what they did was they had serial prostate biopsies. So they actually, at the beginning of the study, these 30 men each had a needle biopsy done of the cancer in their prostate gland. And at 3 months later, they had another biopsy done of the same tissue. So what the scientists collected was small sample of the cancerous cells from their prostate. Now, from those cells, they were able to purify messenger RNA. If you remember your biology, the messenger RNA is the molecule that is used when a gene is turned on. It makes messenger RNA, and the messenger RNA goes out into the cell, and the ribosome turns it into protein. So by doing this, it's sort of like the NSA listening in on whoever might be listening, to the gene transcription. And so the messenger RNA only lasts for a few hours to a few days. So from this, they were able to tell the difference in gene expression, right? Okay. It was a 3-month program. Had a 3-day intensive residential retreat like they could do out at Eden Valley's Wellness Center. They had a low-fat diet, whole foods, plant-based, supplemented with soy, which was a daily serving of tofu, plus 58 grams of soy protein beverage. President Clinton approved of that from what I understand. Fish oil, vitamin E, selenium. That's selenium is easily available in Brazil nuts. And vitamin C, and all food was provided so that these men could stay on the program with no problem. So here's the bottom line. I'm sorry, this is hard to see with the lights on, but it's called the heat map. And what you would find here is there's 30 columns over here of the men before the intervention and 30 columns over here after the intervention. Each row is a different gene that was examined. And the way it works is if there's a lot of messenger RNA, the color is very dark or red. If there's very little messenger RNA, it would be light colored, okay, or greenish or whatever. So you should be able to see that in general, just by looking at this side compared to this side, it's supposed to look lighter on this side. Anyway, it is lighter. And in fact, there was a difference of about 450 genes that were turned down or off. We're only looking at a few here in this, but if you look at all of them, there's over 450 genes in 90 days that were turned down or off, about 50 that were turned up or on. Now, before we get upset about that, it turns out that some of the genes are anti-cancer. You want them up, okay? So this was actually, by and large, this was good, and this was also good. The changes went in the right direction. And in fact, the other thing that they said was on digital examination, the tumor, the size of the prostate was decreasing in size. So there was clinical regression, there was genetic change. Now, I'm going to skip that one. Just the bottom line is that they did a study on sleep deprivation, and they found that going without sleep for a week, getting only, I think it was less than 6 hours of sleep for a week, there was a 711 genes that the switch settings were clearly changed by that. So, you know, it's not just that you're not feeling very energetic the next day. You've got genes that are changed. And exercise was another one. I'm behind on it on my time, so I want to hasten the pace. Let me just show you this one though. This was published in 2004. This is like 10 years ago almost. This is stenting, okay? Percutaneous coronary intervention versus exercise. So this was talking about angioplasty stent versus exercise for someone with stable coronary artery disease. Okay? Coronary artery disease is when your arteries get blocked, plugged up in your heart. And this study, one-year study, how do lifestyle changes compare to stenting for coronary heart disease? It was a randomized trial, one of the best designs we have, to determine whether the stent is as effective as 1 year of exercise in patients with stable coronary artery disease. And what I always like to ask you is, imagine that you had a loved one that had heart disease, and they got into this study, and which would you hope they would get? I mean, would you want them to just get exercise training, or do you want them to get a stent? Don't answer that yet, because you may change your answer. So 51 patients were assigned to the exercise group. They had 20 minutes or more a day on a bicycle, and only 70% of the symptom-free maximum heart rate. And they had a 60-minute a week group aerobic activity. The other ones had assigned, they got the stent and the angioplasty. Okay, this is what they wanted to do to Mr. Charles Kelly when they wanted to do a stent. So here's what this is called a survival chart. It starts off, everybody is fine, nobody had a heart attack, nobody had pain. But as people start having symptoms, this comes down. So the ones that had the stent, at 12 months, they were 70% of them had not had any event. So this we would call the success, right? I mean, no problem. But 30% did. Of those who had exercise training and did the exercise, 88% were event-free. There's a relative difference of 26%. The right answer is you would hope that they trained your loved one to do exercise on a regular basis. Exercise is more effective than angioplasty and stent. It's other studies I could show you don't have time for. It's more effective than bypass surgery. Did you know the thing that is most affected after 5 years, if you have a bypass, and let me first say, anybody here have a bypass? Good. Okay. If so, I would have asked them to see me if they had a different experience. But here's what the average statistics show. 5 years after a bypass is done, there is more impact on the brain than on the heart. And it's bad. It's bad for the brain.
Say that again.
There's more, after 5 years, within 5 years after having a bypass surgery, there will be more negative impact on the brain than there is positive impact left on the heart. Yes, ma'am.
What is the impact?
Dementia, degradation, mini stroke, deterioration of the brain, cognition, memory.
Why do they think that is?
Can we do that at the Q&A?
Sure.
That's a great question. I do know the answer.
Oxygenation?
So, I'm going to. Now, this is the guy that did the research with the Goody Mouse, Dr. Girdle. Look what he has to say. This part I really love. Couldn't say it better myself. Epigenetics is proving we have some responsibility for the integrity of our genome. This is not just something that you get and it's just, you know. You should be thinking, I am steward of my genome. You are. Before epigenetics, genes predetermined outcomes. And now everything we do, everything we eat, smoke, drink, I mean, you can make this list, can affect our gene expression and that of future generations. Epigenetics introduces the concept of, what did he say? Free will into our understanding, our idea of genetics. We knew that it was there, but we didn't understand the science, how it happened. Now we actually know how the science of free choice having an impact on it. So, all right, every cell in our body has a copy of the genome. Different genes are turned on and off by environment. Diet is the strongest impact. The DNA sequence is essentially fixed. You can't change that. But the epigenome, is stable but reversible. So you can change these switches, and they are stable until changed by something else. Lifestyle choices control gene expression. So, change your diet and lifestyle, changes the epigenome. Change the epigenome, changes gene expression. Change gene expression, well, it literally changes you. We can actually change a skin cell into a cardiac cell by changing the epigenome. So the power of this is actually fantastic.